Okay, so after the conformational search was done, we wanted to extract the actual, hopefully the location of low energy structures to try to elucidate the conformational preference of these peptides. So we have those 25,000 conformations. We clustered them using a standard clustering protocol called k-means. And we, from all the 25,000 conformations, we generated clusters of structurally similar conformations. We determined the number of clusters to be 200 for neuromedin K and 100 for vasopressin, which is a bit more restricted because it has a disulfide bond between two sulfurs. And we took as representatives the lowest energy structures from each one of the clusters, and we picked the lowest ones. So I just want to show you some results of the local minima. So you see here the local minima that we got for neuromedin K with, again, the hydrogens were removed for clarity. So you can see the atomic representations. And the magenta and green lines here represent interactions between atoms that stabilize the structure. The magenta lines here are hydrogen bonds, and the green ones are salt bridges. If you see especially this structure here on the top right, looks a lot like a beta hairpin, which is actually a very nice result because we know that amyloids tend to form beta structures, cross beta structures, which are very prone to self-assembly, which is one of the properties of amyloids. You can also see the four lowest minimum energy structures for vasopressin. And you can also see that a lot of them look like beta hairpins. So later we took some of the local minima and we simulated them using standard MD simulation just to see whether these extracted low energy conformations are actually stable over a period of time, not just during the simulated annealing, because the simulated annealing just visits the lowest energy landscape for a very brief amount of time. So if we take one of those local minima and try to simulate it over a period of time in stable conditions, constant temperature, and it still maintains its overall structure, then we can assume that it might actually be stable for a long enough time to generate amyloids. So the local minima that we extracted and that I showed you in the two previous slides were simulated for each one for 20 nanoseconds in 300 K room temperature under constant volume conditions, just to see if their structure is stable. So here is another application of MD to actually test the structural stability of a structure that we modeled before. And indeed, the beta hairpin conformations that we see here for both of the molecules are very stable in simulation. And they were the ones that we selected for further investigation. Now the next thing we want to do after we investigated the conformational preference of the isolated peptides was to conjugate the polymer molecules. So the polymer that we selected is polylactic acid, PLA, which is shown to be very biocompatible and it has a lot of desired properties that can be used for nanostructure design. So we used the 150 residue long tail of PLA that was linked to the C-terminus, the end of the peptide. So we chose 150 residue long polymer to produce the chemical environment of the available literature. There was somewhat of a disproportion in the size of the polymers with respect to the peptides, and in future investigations we're going to actually work with a shorter polymer. So this is a much larger system. 150 residue polymer is actually pretty big. And in this case, extensive conformational exploration like the one that we did with a small peptide is not really feasible computationally. So we actually started from some extended structure and performed MD simulation to see what the conformational preference over a period of time would be. So we had to parameterize the force field for PLA, which was done using quantum mechanics simulations by my collaborators in Barcelona. And there are a lot of quantum mechanics simulations tools to parameterize force field for non-standard molecules. They don't have standard force fields. And we used similar conditions to the ones that we used in the EMD simulations of the peptides. We used a solvent box, an orthorhombic box, much larger one, of course, which resulted in over 500,000 of atoms for each one of the systems. Okay, so it was actually a very, very big system. So we needed a lot more computational resources to simulate it. And we used similar force field parameters generated by Amber, FFO3. Following by minimization, heating, and equilibration, we ran a production MD simulations for 30 nanoseconds. And we saved a snapshot every 2 picoseconds for analysis. So what you can see here in the picture, on the left-hand side would be the initial conditions in which we started to simulate. And the spheres here, the red and white spheres, are the polymers. And CPK representation, ball and stick representation of the protein, of the peptide, and they're conjugated. And on the right-hand side, you see what they look like in the end of the simulation. So what we found out was that the polymer folded as we expected because of the long hydrophobic tail, which actually folded on itself to create a compact structure, which the peptide resides inside. Now, what we found about the peptide is that while the interactions between atoms in the peptides were slightly different than the ones in the isolated peptide simulation because of the different environment, the structural preference of the peptide when it was conjugated with the polymer were actually very similar, which was a very encouraging result. So this is pretty much where we stand right now, or where we stood when we published this work. And what we're doing right now is to pursue several other directions. First thing we want to do is to test the self-assembly properties of the peptide. We found that the peptides are capable of generating stable beta hairpins, which are good starting points for self-assembly. So what we want to do is to simulate how the self-assembly of peptides would look like using MD simulations, with and without the polymers. And we also are in collaboration with an experimental group in the Polytechnic University of Catalonia who are trying to test the properties of systems like this one in a lab. And we're also in parallel testing PLA with a small peptide, phenylalanine, 2 phenylalanines, which is a smaller system, so it's easier to test in a fluoride-based solution, which are the same experimental conditions that our colleagues are using. And we want to correlate the experimental and the simulation results using both classical mechanics MD and quantum mechanics simulations, and get better insight about what we can do with these systems.